welcome to our discussion on diseases and medically important prokaryotes. So we're going to talk about bacteria. We're going to talk about the pathogenesis, epidemiology, and today's topic is going to be gram-positive bacteria. We'll talk about the acid fast and the spirochetes in this section. I'm going to break this up into several videos because I'm having some technology issues, and also there are some nice breaking places between the different diseases. So let's do it. I've given you a table that's going to have the organisms, so the causative agent, the diseases, and then some distinctive features that I would like you to know. I have made you a Quizlet to help you study and prepare for your lecture exam. I've divided them up into gram-positive organisms and gram-negative organisms. And remember, they're structurally different in their cell wall, and both are going to have a cytoplasmic membrane. Gram-positives, remember, they have the one periplasmic space, and then they have a thick peptidoglycan cell wall. This allows them to retain that crystal violet stain in that differential stain called the gram stain. The gram-negatives have cell membrane, periplasmic space, a very thin layer of peptidoglycan, another periplasmic space, and then that outer membrane. And remember, that outer leaflet is where the lipopolysaccharide is that serves as the endotoxin that makes our immune system crazy. Because of this thin peptidoglycan layer, also they do not hold on to the crystal violet, that primary stain in the gram stain. They can be decolorized by alcohol, and then they get the safranin, which is why they appear pink. Gram-positives are going to appear a purple because they retain that crystal violet. So we've divided them up that way. For each of these, we're going to talk about the pathogenesis. So how do these organisms actually cause disease? So their mechanisms, their virulence factors, and then also some information about the epidemiology. Who is affected by this disease? How often? How is it transmitted? Remember, in order for an organism to cause disease, they have to do three things. First, they have to be able to adhere. And remember that pathogens are going to enter either through a break in the skin or through that mucous membrane. Then they need numbers. We are kind of large. So to do damage, they have to colonize. That means they have to be able to reproduce all the while avoiding destruction by our immune system. And then these organisms are also going to make toxins, or through collateral damage from our immune system, they have to harm us in some way. And so these are parasitic relationships. So the gram-positives, first we're going to do it by shape. We'll talk about the gram-positive cocci. So the two genera are Staphylococcus and Streptococcus. There's two very important members, so Taphagus Aureus and Streptococcus pyogenes, that you're going to be like, wow, you're spending all your time on these two because they cause a whole range of infections based on the strain and the location in the body that they're infecting. Then we'll talk about the spore-forming rods, so those two genera, Bacillus and Clostridium. Then we'll add the non-spore-forming rods, Corynebacterium and Listeria. The big six. So Staphylococcus aureus, Streptococcus pyogenes, Streptococcus pneumoniae. We've talked about Streptococcus mutans and their ability to form a biofilm and plaque on our teeth, so I'll just add a little bit more of information on Streptococcus mutans. So I made tables for some of these, and you can find most of the information for Staphylococcus aureus in chapter 22. I've divided up by color because, I just love to organize things. So the first three, these are all going to be skin infections, and they just have to do with the organism and how they get in, if they can get in through a break in the skin, and what different strains do. This other group of diseases have to do with toxins, so exotoxins and also super antigens. That's the reason for the color coding. These little stars, these skin infections that are caused by Staphylococcus aureus, Streptococcus pyogenes is also capable of causing the same ones. Staphylococcus aureus, we've talked about MRSA, and we did a little bit with them in lab. You know that they are a common skin resident. About 1/3, or your textbook, 30 to 40% of humans are going to be colonized with Staphylococcus aureus on their skin or in their nasal passageways. They can be transferred from person to person, and also they can survive on inanimate objects. So you can also transfer Staphylococcus aureus by fomites. So you touch something, then someone else touches it, and they get your Staphylococcus aureus, or the other way around. Keep in mind, we just discussed microbial genetics, and within one species there are many different strains, and so these strains are going to vary in their virulence depending upon what genes they have, what toxins or enzymes they produce. Staphylococcus aureus is non-invasive, meaning it needs an in, so it needs a break in the skin to cause infection. Leading cause, once again, you think Staphylococcus aureus, think skin, because they're salt tolerant. Remember, the term for that is halotolerant. In a lab, mannitol soft agar has 7.5% salt, which inhibits the growth of other things that are not halotolerant. So it's a great media for selecting for Staphylococcus. So Staphylococcus aureus can cause a multitude of hair follicle infections. So when they get into just the follicle, you are going to basically get a pimple. If it goes a little deeper, then we call that an abscess. So skin abscesses, these are just going to be localized collections of pus, and you can see that this tissue surrounding it is inflamed. So it's just inflammation of a follicle, folliculitis, so basically a pimple, but then it can get larger. This is not a nipple, this is a boil. So when it gets a little larger, we call it a boil, or another name for that is furuncle. If it really gets big and kind of spreads a little bit more in this dermis, then it can be called a carbuncle. What you find in the center of that abscess, remember, pus is a collection of dead neutrophils, bacteria, epithelial cells, and skin secretions, so fluid builds up. If this spreads throughout the deeper layers of the dermis, notice now we've got capillary beds and blood passageways, you can get what's called cellulitis. Cellulitis, deeper layers, if it spreads into the bloodstream, remember we get bacteremia, then we have sepsis, so that overall immune response to the bacteria in the blood, and we know from sepsis we can go to septic shock, so we get that drop in blood pressure and disseminated intravascular clotting that leads to organ failure. So cellulitis, you need to worry if someone starts to run a fever, because that means that infection has spread. Impetigo is really common, especially amongst children. Another name for that is pyoderma. Whenever you see pyo, it means pus. So through minor breaks in the skin, the Staphylococcus aureus, they enter, and then you get this honeypot, sort of honey-colored crust from the pus. It is highly contagious. Children often, too, they're very touchy. So through direct contact, but also, once again, through fomites. And insects can even carry it from one person to the next. It is the #1 leading cause of wound infections. Streptococcus pyogenes also causes wound infections, but #1 is going to be Staphylococcus aureus because they are usually resident on the skin. You can clearly look at this and go, this does not look good. It's going to delay the healing. Also, once again, you can get that large abscess formation, and then if it goes deeper, then it can spread into the bloodstream. Never a good thing. And that's just sort of the progression of a abscess. Some of the cytotoxins and those toxic proteins that some strains make. So proteases, those are just enzymes that break down proteins. And in the connective tissue, we have a lot of collagen in the skin. And so that's going to allow that Staphylococcus aureus to spread, and to kind of think of these enzymes that help them sort of burrow and drill through our tissue. So lipases break down fats in connective tissue. Hyaluronidase breaks down hyaluronic acid, which is also a component of connective tissue, allows for spreading. Sidin, remember that means to kill, so leukocidin, that would be killing those white blood cells, and that's why you see a lot of pus associated with Staphylococcus aureus infections. Coagulase is telling you it can coagulate our blood. So it can cause fibrinogen to go to fibrin. This is also a key diagnostic characteristic of Staphylococcus aureus. And if all goes well, we'll do a coagulase plasma test in lab. They have Also, other defenses that help them to avoid our immune system. They have DNases, so they can break those DNA nets that are cast by those neutrophils. Remember, that's their Spider-Man move. Neutrophils are those first responders that get there on the scene of infection. They're not the only ones that can do it. Streptococcus pyogenes can, Campylobacter jejuni, which causes gastrointestinal distress. Another weapon that they have also is they have what's called protein A, and they can bind the Fc portion of antibodies. So they're just good at avoiding destruction. So those are the skin infections that are caused by Staphylococcus aureus, and they can also be caused by Streptococcus pyogenes. So now we're moving into that release of toxins, and Staphylococcus aureus also makes a toxin that can cause food poisoning. Let's watch.
Staphylococcus aureus is a gram-positive spherical bacterium which on microscopic examination appears in pairs, short chains, or as bunched grape-like clusters. Some strains are capable of producing a highly heat-stable protein toxin that causes illness in humans. Staphylococcal food poisoning is the name of the condition caused by the enterotoxins which some strains of the bacteria can produce. The onset of symptoms from food poisoning from this type of bacteria is usually rapid and in many cases acute, depending on the individual's susceptibility to the toxin, the amount of contaminated food eaten, the amount of toxin in the food ingested, and the general health of the victim. The most common symptoms are nausea, vomiting, retching, abdominal cramping, and prostration. In some severe cases, headache, muscle cramping, and transient changes in blood pressure and pulse rate may occur. Recovery generally takes 2 days, but can take longer. Foods that are frequently incriminated in staphylococcal food poisoning include meat and meat products, poultry and egg products, salads such as egg, tuna, chicken, potato, and macaroni, bakery products such as cream-filled pastries, cream pies, and chocolate eclairs, sandwich fillings, and milk and dairy products. Foods that require considerable handling during preparation and that are kept at slightly elevated temperatures after preparation are frequently involved in staphylococcal food poisoning. Staphylococci exist in air, dust, sewage, water, milk, and food, or on food equipment, environmental surfaces, humans, and animals. Humans and animals are the primary reservoirs. It is present in the nasal passages and throats and on the hair and skin of 50% or more of healthy individuals. This incidence is even higher for those who associate with or who come in contact with sick individuals and hospital environments. Although food handlers are usually the main source of food contamination and food poisoning outbreaks, equipment and environmental surfaces can also be sources of contamination from the bacteria.
So this is going to be poisoning through foodborne intoxication. So this comes on rather quickly after ingesting the food. So the way it goes is from the skin of your food handler to the food that is kept warm. The bacteria multiply and secrete their toxin. This is a super antigen toxin. Remember, this is the one that overrides that specificity of the T cell response. They don't really understand the mechanism of it. They just know that it's a super antigen that causes the abdominal cramping, nausea, and vomiting rather quickly. They also know that this toxin is heat stable, so as the foods are kept nice and warm, like I said, just perfect for the Staphylococcus to divide, and then it's ingested. Also, if a kitchen isn't being cleaned regularly too, once again, Staphylococcus aureus can live on inanimate objects. So, yep, this is that reason for the employees must wash hands before returning to work. Just one of them. One other reason. Taphagus aureus, we've talked about the scalded skin syndrome. It is a toxin that's going to go systemic, and it gets a special name, exfoliatin, and it's telling you what it's doing. It is going to cause a separation between the epidermis and the dermis, which causes the peeling of skin. This is something that's going to mainly affect infants because, one, their immune system is not developed yet, and two, neither is their microflora to protect them from these bacteria. Whenever there's a breach in the skin, then that person is always prone to getting a secondary skin infection. It can be transferred by direct contact. Staphylococcus aureus, certain strains, mind you, are capable of causing toxic shock syndrome. There was a spike in toxic shock in the 1980s due to super absorbent tampons. So the Staphylococcus aureus, from insertion of the tampon, would grow inside the tampon and release a super antigen toxic shock syndrome toxin 1. So you get fever, vomiting, diarrhea. It came with a rash, and you get this massive cytokine storm, and in some instances it led to death. You can also get this through wound infections, post-abortion, or childbirth. But like I said, the big spike occurred in the 1980s. They have since changed the way they manufacture tampons, but the warning label is still there. And so, like I said, just to kind of remind you, we talked about those super antigens and how they cause chaos because they stimulate a whole army of T helper cells. So Staphylococcus enterotoxin, the toxic shock syndrome toxin, and exfoliative toxin, those are all going to be examples of those type of super antigen toxins. Yep. So also food poisoning. And exfoliative. This was an example of their defense. They also have that protein A, so my slide was sort of in the wrong spot. But protein A on the surface of Staphylococcus aureus, it binds that Fc portion of the antibody. Remember, antibodies are supposed to bind specifically to their antigen, and then they are neutralized and targeted for destruction. So it fixes complement, acts as an opsonin. And so Staphylococcus aureus some strains can avoid destruction that way. I think we watched this already, but you know that there are antibiotic resistant strains, so I'm going to leave that one for you. But yeah, so a long time ago, about 70 years ago, Staphylococcus aureus, they were all susceptible to penicillin. They are no longer susceptible to penicillin. Most of them are penicillin resistant, so they have a resistance plasmid that allows them to break what's called a beta-lactam ring. So there's a structure of penicillin, cephalosporins, and then any of the other drugs that are synthetically made versions of penicillin. And so they can break this ring, and that inactivates that antibiotic, so that's what makes them resistant. We talked about methicillin resistant and how some people carry those, and they're very difficult to be killed by antibiotics. It doesn't mean that they're more virulent, it just means that once someone has an infection, it's hard to cure them of that infection. This one now is becoming lower in the hospital community because they're screening for it, and is found out in the community at large. And so affects usually children and young to middle-aged adults. They can just pick it up through a minor wound or a locker room, so often occurs in athletics. A growing threat, though, in the hospitals is going to be vancomycin-resistant Staphylococcus aureus. So usually if someone has MRSA, they use vancomycin as an antibiotic to treat that. And once again, then you're selecting for those that are resistant to it. They thrive, and then they are spread. So this now is not at the community at large like MRSA is. It's in hospitals. So it is classified as a serious threat by the CDC. So here's a table of Staphylococcus aureus. Staphylococcus epidermidis is a common skin, because you can see epidermis is in the name, commensal, so lives on our skin, but when it enters a wound, it can also cause those wound infections. It's just not as virulent. It doesn't have all of those toxic proteins that Staphylococcus aureus has, but it can also cause wound infections. They also have the ability to form biofilms. I've left that there for you. And I'm going to stop this video because I'm just afraid my computer's just going to lock up on me. Come back for part 2, Streptococcus pyogenes.